Interview questions and answers for Pharma, Quality Control, Fresher. Question 1, what is pH? The pH is the negative logarithm of the hydrogen ion concentration. Question 2, what is pH range? The range goes from 0 to 14, with 7 being neutral. pHs of less than 7 indicate acidity, whereas a pH of greater than 7 indicates a base. Question 3, what is 5-point pH calibration? Perform the 5-point calibration using standard buffers of pH 1.68, 5.00, 7.00, 10.01 and 12.45. Question 4, what is solubility? Solubility indicates the upper concentration a compound reaches in a solution. Solubility is a very important property in drug discovery and development, because concentration affects so many aspects of pharmacology.
Question 5, what is thin layer chromatography? Thin layer chromatography, TLC, is an affinity-based method used to separate compounds in a mixture. TLC is a highly versatile separation method that is widely used for both qualitative and quantitative sample analysis.
Question 6, definition of normality? The number of gram or mole equivalents of solute present in 1 liter of a solution. Question 7, definition of molarity? The number of moles of a solute dissolved in 1 liter, 1000 milliliters, of the solution is known as the molarity of the solution. Question 8, what is Karl Fischer titration? Karl Fischer titration is a classic titration method in chemical analysis that uses coulometric or volumetric titration to determine trace amounts of water. Question 9, what is loss on drying? Loss on drying, LOD, is determined by heating the sample below its melting point in an oven, and it includes all volatile matter including water content and solvents. Question 10, what is principle of UV spectrophotometer? The principle of UV visible spectroscopy is based on the absorption of ultraviolet light or visible light by chemical compounds, which results in the production of distinct spectra. Question 11, what is the range of UV visible spectroscopy? It is 400 to 800 nanometers. Question 12, what is the range of ultraviolet spectroscopy? It is 200 to 400 nanometers.
Question 13, what is infrared spectroscopy? Infrared spectroscopy is the measurement of the interaction of infrared radiation with matter by absorption, emission, or reflection. It is used to study and identify chemical substances or functional groups in solid, liquid, or gaseous forms. Question 14, what is the range of infrared spectroscopy? The infrared range covers 700 to 1000 nanometers, wavelength, or 14,286 to 12,800 centimeters -1, wave number, and ultraviolet radiation has wave numbers above these, approximately 25,000 to 50,000 centimeters -1, or 100 to 400 nanometers, wavelength.
Question 15, what is pharmacopoeia? A pharmacopoeia is a legally binding collection of standards and quality specifications for medicines used in a country or region.
Question 16, what are types of pharmacopoeias? United States Pharmacopoeia, British Pharmacopoeia, European Pharmacopoeia, International Pharmacopoeia, Japanese Pharmacopoeia, Indian Pharmacopoeia, Chinese Pharmacopoeia. Question 17, what is dissolution? Dissolution rate may be defined as amount of drug substance that goes in the solution per unit time under standard conditions of liquid/solid interface, temperature and solvent composition. It can be considered as a specific type of certain heterogeneous reaction in which a mass transfer results as a net effect between escape and deposition of solute molecules at a solid surface.
Question 18, what is HPLC? High Performance Liquid Chromatography, HPLC, is an analytical technique to separate, identify, and quantify components in a mixture. Question 19, what is mobile phase? The solvent used to separate components in a liquid sample for HPLC analysis is called the mobile phase.
Question 20. What is analytical method validation? Analytical method validation establishes documented evidence that the procedure adopted for a test is fit for the intended purpose in terms of quality, reliability and consistency of results. Question 21. What are the parameters of method validation? Selectivity and specificity, linearity, range, accuracy, precision, limit of quantification, ruggedness, robustness,